All right. Well, thank you. Um, so I have to admit, when I give a talk, I usually know my audience a bit. I'm familiar with, I have a sense of who's going to be there and why they're there and whatnot. And I don't have that here. So I don't really know who's listening or why you're listening. But I thank you for the invitation and I thank you for coming. But that really affects the way I pitch the talk or structure the talk. So what I've done here, just to start out by saying that I've made the talk reasonably broad, just so you can see the context of where the work fits. But if it's not broad enough or too broad, if I skipped over too much of the chemistry for whatnot, then please interrupt me. Let me know. I'm happy to explain anything. I'm happy to be interrupted at any time and take the talk in a different direction. So, as you just heard, I'm really an organic chemist. I love organic chemistry, and I really don't do metals. My whole life I stayed away from them. But then about 10 years ago, a little more, for reasons I'll explain during the talk, I got pulled into looking at organometallics. And so it took many years to develop the methods that I'm going to describe in the talk. But this whole issue of iron limitation in the twilight zone, and the twilight zone in the ocean is just that region right below the euphotic zone, right where light is sort of going away, and we're at the top of the deep ocean. So there's this layer in there between, let's say, 200 meters, which is the bottom of the euphotic zone, to up to 1,000 meters, where the ecosystems are really transitioning. And this issue of iron limitation there was something that we weren't looking for. It sort of came up. We've just finished the actual research or analysis that we're going to do for it, and we're about to submit it for publication. So in one sense, this talk's a trial balloon. You're my test audience, and so any comments you have about it would be really helpful to me, because as I said, we're about to submit the manuscript, and any input before that goes would be helpful to us. Okay, all that said, let's see, how do I? All right, just some background into the problem. So about 40 years ago, in the mid-1980s, geochemists had gotten to the point where they could finally make really high quality measurements of dissolved iron in the ocean. And up till that point, there's two real major problems here. First was that it turned out the iron concentrations were really super low, were picomolar, and just developing the analytical methods that could reliably measure concentrations that low at high precision was a real challenge. And then the second challenge was that iron contamination is really prevalent everywhere in the labs. We go out to sea and we're sitting on a ship made of iron. And so actually to collect the samples cleanly and to analyze them cleanly took many, many years to work out all those issues. But that had been largely resolved by the mid-1980s. And what geochemists found was, where's my pointer? Okay, I hope you can see the pointer, was that the profile of iron in the ocean looks something like this: very low concentrations in the surface. Well, this is nanomole scale on the top. So in this case, maybe 100 picomoles, 50 picomoles in the surface. And then once you get through the euphotic zone, really a rapid recovery of concentrations down to about 1,000 meters. And then things look fairly stable below that. So this is the classic nutrient profile. And the explanation is simply that ocean biology is removing iron from the water column, drawing the concentrations to really low levels. And then when you get below the euphotic zone, below the zone of primary productivity, down here, what's happening is particles are sinking out of the euphotic zone. They're carrying their iron, their carbon, their nitrogen, their phosphorus with them. They're degraded by heterotrophic microbes. And as part of that degradation product, iron is being resupplied back into the dissolved phase. So that's, we see these profiles like this for nitrate, for ammonia, for phosphate, for really key nutrients for phytoplankton. And they all look like this, as well as other trace metals that are used in biology. The other thing that was realized was from laboratory culture experiments, in some places of the ocean, the iron concentrations were really at the limit that phytoplankton can grow at. So they were able to grow, to manipulate cultures with iron concentrations down into the picomolar range. And when iron got so low, below a few tens of picomoles, even the smallest and most efficient photoautotrope were unable to grow, certainly at maximal growth rates. So it looked like biology was pulling concentrations of iron down to sort of the limit that they needed to thrive. The other advance that happened about this time was that we got really the first global scale and seasonal models of sea surface chlorophyll in the ocean. And this is data collected by satellite. They've been collecting it for more than a decade, for almost 2 decades at this point. But finally, what came to fruition was the algorithms that were needed to take this optical data, turn it into surface chlorophyll, validate those measurements, and put those into large-scale models of global surface chlorophyll of the ocean. And chlorophyll is a proxy for autotrophic biomass, which is a proxy for primary production. So we get these maps of chlorophyll, of biomass, of primary production. There's a lot of ingredients that goes into making that sausage. But the output was that certainly there are large areas of the ocean here in dark blue. So the scale is, the cool colors are low concentrations, the hot colors are high concentrations. But you see through many of these really large ocean gyres, the North Pacific and South Pacific, North and South Atlantic, Indian Ocean, very low concentrations of chlorophyll. Whereas in other areas, certainly coastal areas and some of the very high latitude areas seasonally, you had very high concentrations of chlorophyll, ergo biomass, ergo productivity. The other advance that happened at this time is we had a fair amount of data of ocean nutrient distributions, and this data was put into global circulation models and allowed us to take a really global look at sea surface nutrient values. And that's shown here on the right. This is from a paper by Bristow et al., I should say, where again, the hot colors are high concentrations of nitrate and the cool colors are low concentrations. And where we saw low, standing biomass, we see low concentrations of nutrients. And then, not surprisingly, where we have some high amounts of chlorophyll or biomass, we have higher nutrient concentrations. But what was realized, and this had actually been known from ocean observations, there were portions of the ocean, and I circled here sort of at the equatorial Eastern Pacific, and certainly throughout the Southern Ocean, where we had really high concentrations of nutrients, but reasonably low concentrations of biomass. And these are called high nutrient, low chlorophyll regions. And the thinking was that if you have sunlight and you have nutrients, the growth of bacteria, of autotrophs, of algae and cyanobacteria and the like, can easily pull down nutrients to limiting concentrations in the dark blue here. But there were these regions of the ocean where we still had high stocks of nutrients, but very low biomass. And most notably, you can see throughout the Southern Ocean, there's regions here, certainly in the Eastern Pacific, where we have, micromolar concentrations of nitrate, but very low standing stocks of biomass. And this sort of sparked a debate about why these high nutrient, low chlorophyll regions existed. There were different explanations for that. It's about grazing efficiency, about growth rates of phytoplankton. But some geochemists noticed that in these areas, iron concentrations got really, really low, in the tens of picomolars. And they hypothesized that iron was also a limiting nutrient in the ocean. Up till that point, sort of phosphate and nitrate were seen as the key nutrients that determined marine productivity. But it was postulated, it's called the iron hypothesis, that iron was also a key nutrient. And in these regions where iron was super low, that would limit phytoplankton production, and that they wouldn't be able to grow, and you could still have high stocks of nitrogen and phosphorus. And so that led to a number of laboratory experiments of people looking at phytoplankton growth in cultures. And most well-known are these mesoscale iron fertilization experiments. And so what you do is you go out on the ship, that's this picture on the upper left, you spray iron into the ocean, you can see this rust-colored trace trailing the ship, and then you wait a little bit, and then you start measuring the productivity inside and outside that patch. And you can see that here. This is a satellite image from one of these experiments in the Southern Ocean. It was done in an eddy, so to speak. So it was fertilized here, and the water moves this way over time. The scale is chlorophyll now. Again, red colors are high chlorophyll, blue are low chlorophyll. And you can see where it's been fertilized, you get really high standing stocks of chlorophyll A and productivity. So it was a real direct demonstration that there were lots of areas in the ocean that were actually limited by iron. And then if you would add iron, you get abundant phytoplankton growth. And you can see that here in the lower right, where this is from a Japanese experiment in the Northwest Pacific Ocean. This is a phytoplankton, a net toe inside one of these blooms, artificial blooms. And here's a companion net toe outside of the bloom, and you can just see the difference in biomass. And now these have been done maybe 15 or 20 times in the different HNLC regions of the ocean. And repeatedly, they show that when you add iron, you can make phytoplankton bloom, so that there are these regions of the oceans that are iron limited. So all that data was put into models, and these models relied on the distribution of the nutrient concentrations in different water masses, and then the nutrient requirements of different types of photoautotrophs. So the elemental ratios of iron to carbon to nitrogen to phosphorus. And this is an output of one of those models from Moore in 2004. It's a map of the ocean, and it's color-coded to the limiting nutrients. So most of the ocean is purple, and it's probably, the phytoplankton communities are probably limited by nitrogen, but large swaths of the ocean are colored in blue, and they appear to be limited by iron, at least in terms of what the model output suggests. And so something like 30% of the ocean is thought, surface ocean is thought to be iron limited. So just as a little sidebar, because you might have heard all that before, right away, even in the 1990s, people were cognizant of the fact that we're headed for a climate crisis because of greenhouse CO2 in the atmosphere, and this was proposed as a way to mitigate that. And the idea is that you could, just like they did in those mesoscale fertilization experiments, put chips out in the ocean. They could spray iron into seawater that would stimulate a bloom. That bloom would pull CO2 out of the atmosphere. The bloom would eventually die and sink, get consumed by heterotrophs, or just sink on its own. And that carbon that was incorporated into the bloom, some fraction of it would be transported into the deep ocean. So it's a way of moving CO2 from the atmosphere and sequestering it in the deep ocean. And the attractive aspect of it was that the ratio of iron to carbon in phytoplankton cells is like 1 to 10,000 to 100,000. And so with a little bit of iron, you could potentially sequester a large amount of carbon. And of course, there's pros and cons to this. This is from an article in Discover magazine. So this was debated in the 90s, and then it sort of died out, and then it had a new life in the early 2000s. Then it died out because the cons are really scary. That was said. But recently, now there's a lot of talk, at least in the US, about active carbon disposal or removal from the atmosphere and how we're going to do it. So this is from just an opinion piece from the New York Times, which is a paper of record in the US. It has the largest circulation and often has the most interesting articles. But it's an essay from September 14th, last month, essentially arguing that we could use iron dust to reverse the course of climate change. It's by some marine scientists and some modelers. And, they argue that, as crazy as it might sound, adding iron to the ocean to fertilize the ocean might be the most effective and affordable way, not just to slow global warming, but to reverse its course. So I don't agree with the article, but this is just a really important issue being debated now. we've been told by the government that they're really going to investigate ways to recover CO2 from the atmosphere. And so now there's a large group of marine biogeochemists who have jumped on this and are going to start working on this problem again. So that's sort of the background of this. There's one other really key fact that I need to discuss, and that is, even though the concentration of iron is really low in these iron-limited regions, they're still 100 or 1,000 times what they should be based on just chemical thermodynamics. So if you calculate the solubility of iron-3 in seawater, it should be a picomole or less. And even in these really low iron regions, we find that the concentrations are tens to hundreds of picomoles. So iron is much more soluble in water than it should be, in ocean than it should be. And the reason for that is that it's a very sticky element. And all that iron is basically stuck to organic matter. And so organic matter certainly keeps iron in solution, but it's been known for a long time that it also affects its bioavailability. And this goes back to that model I showed you earlier, where they were using models of elemental quotas in phytoplankton and the distribution of nutrients in seawater to see what parts of the ocean was iron limited versus nitrogen versus phosphorus. And the assumption there was that all of the iron was bioavailable. But in fact, we know that that's not true. And we know that the organic matter that keeps the iron in solution really impacts its bioavailability. And you can see that very nicely here in an experiment done by Mark Wells around 2000. And what he did is he just took coastal seawater, he added some radioactive iron to it to look at iron uptake, and he added some C14 bicarbonate to look at carbon uptake. So his control is just the seawater. His experimental is he has this compound DFB. It's a very strong chelator for iron. So it grabs iron, it holds it really tightly. It's something that we get out of fungi that grow in soils. And you can see that in his experimental treatment. So there's this same experiment with just two different size classes of microbes. He filters it through a greater than 5 micron and less than 5 micron. So you can see when he adds this DFB, the iron uptake in both size fractions, the large and small size fractions of photoautotrophs, goes way down. It's only 3 or 4% of the control. And likewise for carbon uptake, for carbon fixation, it's also only a small fraction of what it is in control. So papers like this argue that the form of the organic matter that binds the iron is really key to whether the phytoplankton can use it. And so that led to the question that is still being debated, is like, how do microbes in the ocean acquire iron? And this is something that's been studied in soils for a long, long time. Even though soil has a lot of iron in it, most of that iron is not bioavailable to plants, but plants require it. So one of the strategies that's been developed by microbes living in soils is they use these, they synthesize these compounds called siderophores, which just means iron-loving. And what they are is they're small metabolites. They bind iron really strongly. They have these stability coefficients of 10 to the 30 to 10 to the 50. They secrete these siderophores into the environment. The siderophore will bind iron from even strong iron minerals. And then the cells have these specialized transporters that will recognize the siderophore iron complex, bring it into the periplasmic space, transport it into the cell. And in the cell, there's a number of different ways to recover that iron. One is just a simple reduction by a reductase. It takes the iron 3 to iron 2. That dissociates from the siderophore, and the siderophore is pumped back into the environment to grab more iron. So it's a small organic compound. It's released, it grabs iron, it's transported into the cell, the iron's recovered, and it's sent back out into the environment. And so there's been many, many microbes isolated from soils that do this. There's something like 400 different known siderophores. They all have somewhat similar chemistry, or a small a narrow range of chemistries, but they're really designed to bind iron. So this makes sense in soils because you can imagine the bacteria is sitting on a soil particle or piece of organic matter or mineral. It can excrete the siderophore, and it's going to be right there. It's not going to go anywhere, and it can take it back up. In seawater, you have a really different situation, though. It's a very dilute environment. This is where I guess microscale ocean physics comes into it. It's a very dilute environment. It's a very turbulent environment. And it was argued for almost 20 years about whether siderophores made sense in an aquatic environment, right? So there were a number of studies looking at the energetics and efficiency of uptake. And they were just models that would go, well, to make a siderophore takes a lot of energy. Then you've got to release it into the environment. And as soon as it's gone, you may never see that siderophore again. And so really, how much iron can you acquire from this method? And is it worth it in terms of the metabolic energy that's been put into it? And repeatedly, the models suggested that it wasn't a great way to get iron from the environment. And where it might work in soils, it really wouldn't work in the ocean. At the time, well, this is still true today, a lot of these siderophores have been discovered in cultures that you would then bring to iron limitation and look for siderophores being produced. Siderophores are well known in freshwater systems. There's a number of freshwater cyanobacteria that are known to make siderophores. Even today, there's only one marine cyanobacterium, and it was isolated from sediments in a fish pond in Puerto Rico. So people debate whether it's really marine or not. But only one cyanobacteria that's even quasi-marine is known to make siderophores. And finally, again, at the time, sort of in the late 1990s, early 2000s, we had the ability to sequence whole genomes for important key marine phytoplankton and look at metagenomes in the ocean. And we really, at the time, couldn't find any genomes in the major groups of marine cyanobacteria that either made siderophores or had the ability to take them up. And so there was some suggestions from the chemical side of things in terms of the iron concentrations, the organic matter complexation iron, that would argue that siderophores were probably out there. But there was a lot of other counter arguments that suggest that they weren't really effective in the marine environment. So, there was this debate about, do marine microbes use siderophores to acquire iron? And I had been aware of that debate for a long time. I really didn't want to get involved, but for a number of reasons, I eventually did. And the problem was, so the answer is to go out and to analyze siderophores in seawater. The problem is that that's really tough to do. And so to illustrate that, I've just drawn this circle here, which is if you were to take a liter of seawater and just evaporate it, you'd get 35 grams of salt and you'd get a milligram of organic matter. So I tried to scale the area of the little red dot to the gray circle to show you how much salt versus organic matter you get. So that's one problem, is all the salt. And then the second problem is, even if you then zoomed into that organic matter, made that the size of the gray dot now, the iron ligands we knew from just iron concentration were picomolar. And so now they're the size of the dot. So you're proverbially trying to find the needle in the haystack. And then on top of it, we have this issue of contamination and whatnot. So there were no really good ways at the time to do this. And so that's what we set out to do. And so, I should say, the complication to this is that, we have mass spectral data on the organic carbon, and it turns out there are tens to hundreds of thousands of different organic compounds in dissolved organic matter in the ocean. So this is a typical mass spectrum that you get if you just isolated some organic matter and looked at the masses now. So this is mass range versus intensity. And you see there's just thousands and thousands of peaks. And you don't know which of those masses corresponds to the few compounds that have iron in them. So that's what we set out to tackle. And so the first step was really easy. It's been done for decades. it's well known, is you can just take seawater, is to get rid of the salt. You push it through a column, which is just a little container packed with engineered plastic beads. The water and the salt goes through. The organic matter that's in the seawater will stick to the plastic beads. After you've collected your sample, you can flush it with fresh water that gets rid of any remaining salt. And then you use organic solvents to recover the organic matter into a beaker. So it's a way to concentrate the organic matter, which is here shown in little colored dots, into a small sample. And on the right here, I've just shown what these things look like. They're plastic tubes. Down here is the engineered beads. We push the seawater through, and you can see it turns brown and orange. And that's all the organic matter being concentrated on the column. And then we recover that. And the nice part about this is if we looked at the 400 known siderophores, you can predict that nearly all of them will stick to this and can be recovered from seawater by this technique. So that gets around the salt problem. The next problem was actually to find the iron compounds in that mixture of tens or hundreds of thousands of compounds that are in organic matter in seawater. So we do that in 2 steps. So the first step is we just take the sample here, all these different colored dots, and we use chromatography to separate them. So this is essentially just a much more sophisticated way of doing this extraction. It's again a metal tube or a plastic tube in our case, packed with engineered plastic beads. The sample goes in, all the compounds are mixed up, and you pump solvent through this, and the compounds will migrate down the column, but they'll do so at different rates. And so what starts out as a mixture, at the end, you sort of separate the beads here by color, the different compounds shown in different colors, and they exit the column at a very specific time, and they all exit together. So it's a way to take a mixture of things and separate it into purified components. When it comes out of that column, we put it into an inductively coupled plasma mass spectrometer. And so this is a mass spectrometer. We have an oven in it that's heated to something like 8,000 degrees. We put in oxygen and we essentially burn everything. The carbon goes off as CO2, but any metals and whatnot in the other elements, phosphorus as well, get burn into their elemental form, and we charge them and we place a charge on them, and it goes into a mass spectrometer. And you can tune that mass spectrometer to look at whatever mass you want. And so here's the output of that system. So this is now the bottom axis is time. So that's the time it took to go through that chromatography column and come out in the detector, which is the mass spectrometer. And this is intensity on the vertical axis, but this is iron 56. So all you're looking at is the sort of number of iron atoms coming out of that separation versus time. And you can see there's sort of a background, and then we get these peaks, and these peaks represent organic compounds leaving that column that have iron in it. And so what this allows us to do is it allows us to quantify how much iron is in any given organic compound. And it allows us to know in that separation when that iron compound exits the column. And so I should say that, there are tens or hundreds of thousands of different compounds in that mixture. That separation, these peaks have peak widths of a few seconds, and the whole run takes place in an hour. So it cuts down the number of compounds coming out at any given time, but it's not a pure compound. Even though these look like very pure, peaks and very pure compounds, there's lots of other stuff in that, but you're only seeing the iron. So you're really just seeing where the iron comes out. So that tells us where it is and how much of it's there. And then the second thing is we put it through a different mass spectrometer. So we split the output, or else we do it separately. Sort of the same separation again, but now it's going to a sort of conventional electrospray ionization. This doesn't destroy the compounds. They just get ionized. They go to a detector. The detector scans for mass. So here's what the output looks like. Here's my retention time. And we're collecting spectra all during this analysis. And so using the first data from the iron, we know that at 38 minutes, there's iron in there. So we can pull out the spectrum, let's say here at 38 minutes, and then we can look at it and we can try to find the iron. So here's the spectrum in the background. And the way we find the iron is we wrote algorithms. And what the algorithms do is there are two isotopes of iron that naturally occur. One is iron 56. It has a mass of 55.9349. And then there's iron 54, which has a lower mass, and they differ by 1.995 daltons. And we also know their relative abundance based on their crustal abundance is 92:6. So the algorithm will go through each of these thousands of peaks. It gets the mass, and it asks, is there another mass that's 1.995 daltons greater, let's say? And if there is the intensity ratio what we expect it to be? And if the answer to both of those questions is yes, that mass is pulled out and put to a mass list, because those two different compounds have the right mass to have these right elemental combination and the right relative intensity. And so here's an example of that. Here's my iron chromatogram on top. This is from a culture of Pseudo-Alteramonas, I should say. And we have these different peaks. And we set the algorithm loose on this peak at 38 minutes, and what it came out with was a mass at 985.405, and when at lower mass, it took the lower mass peak, it scales it by 15.7, which is the ratio, and you can see the two masses plot right on top of each other. The lower mass element is scaled to its appropriate height. And you can see that they just perfectly overlay each other. So the algorithm allows us to look at this peak and find out what the mass, the molecular weight is of the two isotopes of iron in that peak. And then we can compare that with lists of known siderophores. And that's what I'm showing here. So here's my output from the culture, and here's my output from the algorithm. And we can see these different masses that it finds for the different iron ligands that are in here. And it turns out there are a series of rhinobactins. These are known siderophores that are made from a bacteria that was isolated, I think, from an oil spill. So that's how it all works. And so the next step was to go out into the ocean. So we weren't funded at the time to do this because we were actually told that it can't be done. So we got together with some colleagues, and they were going on this cruise from Peru out to Tahiti. And this crosses one of those H&LC regions in the equatorial Eastern Pacific as part of the GEOTRACES program, which is this international program with many, many different countries involved in this to make really high resolution and high precision maps of trace elements in the ocean. And so we weren't part of that. But we snuck on the ship, we installed some sampling equipment, and we were able to get 5 or 6 samples just from the surface water. So the ship has a seawater intake, and we put our sampling gear on that. And so we were able to collect a small suite of surface samples here along the cruise track. And then below this, I've plotted the nitrate and the iron. And you can see there's this H&LC region between, about 85 and 100 degrees west latitude. So lots of nitrate in surface water. This is a plot of 0 to 90 meters, the euphotic zone. And you see when you get out to about 80-something degrees, you still have lots of nitrate, but iron has gone away. So we have lots of nutrients, low iron, and then beyond that, low nutrients and low iron. So here are these chromatograms that from the coastal region, from this H&LC region, and from the oligotrophic. Here's my retention time on the bottom versus intensity. The red trace is iron 56. And you can see in the coastal region, we see a lot of different peaks here, but they're relatively low concentrations. When we get out to this H&LC region, the peaks change and they're much more abundant. And then when we go out to the oligotrophic, they mostly go away. So the red trace is the iron, is what we sense by that iron 56 trace that I showed you, the ICP-MS. But if you remember back to the way I described how siderophores work, the microbes makes the siderophore will put it out in the environment, it bumps into an atom of iron, it grabs it, and then that's transported back into the cell where the iron's recovered. But then it takes that siderophore and puts it back out into seawater. So we thought, well, maybe there's a lot of siderophores in seawater that don't have iron in it yet. They haven't bumped into an iron atom to extract it. So what we did is, after we analyzed the sample, we added iron to the extract. And then we compared the trace after the iron added with the original. And I can't really align these easily, but if you do align these two and bring the baselines together, you see there's very little siderophore without iron in the coastal region. There are siderophores there, they're low concentrations, and they already have iron pretty much. But you can see there's a huge difference in these HNLC regions. There's a lot of peaks here, and the intensity of the peaks are such that there's a lot of siderophore out there that hasn't bumped into the iron yet. And it suggests that this thing is happening really fast, right? Because we did calculations about how quickly that siderophore should bump into an iron and have iron in it. And that process is pretty quickly. And the fact that we have a fairly high steady state concentrations of siderophore without iron means that this whole process must be going on quickly, because we can build up a reasonably high concentration of iron-free siderophores in seawater, which is competing, their production is competing with the iron complexation. And in the oligotrophic, again, that isn't quite true. So this was a very nice demonstration that, first, the siderophores are out there. Secondly, that they change with the different environments. The concentrations are fairly high. They're tens of picomoles, and that there were siderophores in seawater that didn't have iron in them yet. So it was probably a very rapid process. So just quickly to compare, these are the siderophores that we found in Ultramonazius marinobactens. The one we found in the tropical Pacific were related structurally. They're amphibactens. They're different. They're made by similar biosynthetic pathways, but they all have this sort of part of the molecule that complexes iron, and a part of it that is just a fatty acid tail. And in culture, what happens is that this tail anchors the siderophore on the cell into the cell membrane, so it doesn't diffuse away easily. And the idea was that this is a way to bypass that problem with, releasing a siderophore into the environment, and then you'll never see it again, which will reduce its efficiency. So the thinking was that when we first looked at that, we go, well, these things are probably membrane associated. They're sort of sticking out there. They get the iron. They're pushed back into the cell and then pop back out. And that would bypass a lot of the problems with siderophore use in the marine environment. That turns out not to be true, but that's what we were thinking. And just a little sidebar on that. Because we knew which siderophores were out there, we could look for their biosynthetic pathways. So we used the the Tara Oceans Metagenome Catalog. This is this large effort funded by the European Union to actually recover genomes from all the different ocean basins. And when we did a search of those genomes, we can find that these amphibactens were actually quite common in seawater in the South Atlantic, in the Indian Ocean, in the South and North Pacific. So it looked like siderophores were being produced all over the ocean. So after that, we were actually funded now through GeoTraces to look at the distribution of siderophores across different ocean basins. And so this is the East Pacific. This is the GP15 cruise, which was in 2015 probably, or 17. So we sampled, we did full water column profiles from all these stations between Alaska in the north and Tahiti in the south. We crossed different ocean provinces. So the background is this ocean color image. So the higher, the lighter colors are higher chlorophyll. The lower colors are these oligotrophic gyres. We went through coastal systems, HNLC region here, oligotrophic gyre, another HNLC region, another oligotrophic gyre. And our goal was to actually look at the siderophore distributions in those. And what we expected was that, when we get in these HNLC regions, we'd see high concentrations of siderophores. When we went into the gyres, which have relatively high concentrations of iron, low concentrations of other nutrients, if you can remember that plot I showed of surface nitrate in the global ocean, there are these dark, blue patches. Those are these oligotrophic gyres, and we expected to find very low concentrations there. And in the surface water, so this is a plot of 0 to 1,000 meters. The euphotic zone is in this upper 200 meters. It varies with location. But you can see in the HNLC regions, in the euphotic zone, we found high concentrations of siderophores. So the color is, hot colors are high concentrations, low or low concentrations. So in these HNLC regions, we find our siderophores in the surface where we expect them. In the oligotrophic gyres, we really didn't find many siderophores, and we didn't expect them there. But the real surprise was that we didn't expect at all was really most of the siderophores were right below the euphotic zone. And it was a surprise because, rates of production are really high in the surface. And then when you get below the surface, you're into heterotrophic production. And all that production is being fueled by sort of the downward flux of sinking organic matter. And so rates, temperature drops, the substrate, the food supply drops, because in the surface, you have all this autotrophic production. You've got light, you've got nutrients, lots of production of organic matter. Once you get below the euphotic zone, the supply of carbon drops, the temperatures drop, the rates of bacterial production drop. We know all that. And that's right at the zone where nutrients come back up. So if you remember the profile of iron in one of the first slides, euphotic zone really low, and then below that, it goes up very quickly. So the concentrations of iron we thought would be higher here, the rates would be lower, and we really didn't see any reason why anyone would be limited by iron here. So we were looking at that data. So these are different ways to think about that. Our first thought was that, low iron, then you have iron stress or iron deficiency, iron limitation, and then you make siderophores. So we plotted, this is dissolved iron here on the horizontal axis versus concentrations of siderophores on the vertical axis. And you can see it's just a scatter plot. It really doesn't show any correlation between iron concentrations and siderophore production. Then we looked at sort of the iron to nitrogen ratio. So this is nitrate, which is the principal form of the nitrogen nutrient. And what we noticed, it isn't like there's a one-to-one correlation, but we noticed that most of the siderophore production where we had higher values of siderophores occurred below iron to nitrogen ratio of 50. That's what the dashed line is here. And this is from a paper from Philippe Tortel, which is one of the very few, like 2 or 3 papers that measure the iron to nitrogen or other elemental ratios in bacteria that were grown under iron-limited conditions. So you're a heterotrophic bacteria, you're given lots of substrate, but the iron concentration has been knocked down. And in the biomass that's produced under those conditions, the iron to nitrogen ratio is 50 micromoles per mole. And we noticed that most of the high concentrations of siderophores were in waters that had nutrient ratios below that. And so our thought was simply that it shows that these were probably iron limited, that these were iron limited bacteria. And the reason why they were showing that limitation is because they didn't have enough iron to consume the nitrogen that was available. And so if you take that iron to nitrogen ratio of 50 micromole to mole and you plot it over our section here, this is again a plot of siderophore concentrations. The scale's a little different, which is why it looks different. You can see that in the regions where the iron to nitrate ratio is higher than that, we only occasionally see siderophores. And it's right at that boundary where that 50 micromole per mole boundary is where we see these high concentrations of siderophores in the mesopelagic. And so this is a plot of just that data from station 14, which is here in the North Pacific subtropical gyre. The red plot is the siderophore concentrations. The yellow is sort of the nitrate. And you can see just when nitrate both values are very low of iron and nitrate in the surface. But when nitrate starts to come back in, we see this really big boom in siderophore production that peters out with depth. And our thinking is that you have lots of carbon sinking into this region, so there's lots of substrate. That peters out as you go deeper. You have plenty of nitrogen to use that substrate, but you just don't have enough iron. And so that stimulates siderophore production. Just a few side comments. When we look through that section in the North Pacific now, we see different siderophores in different places, which is what we were hoping for. And then the other thing that we didn't expect, so here's iron on the top on one sample, and here's the trace of aluminum on the bottom, and you can see they're almost identical. And aluminum-3 can substitute for iron in siderophores. It's not bound as strongly, but there's a lot of aluminum in seawater. And again, this sort of indicates that these siderophores are being pumped into the seawater by the microbes. They can bind iron, but if they happen to bump into an aluminum atom first, they'll take the aluminum atom. And that's why we get these traces of aluminum that look just like iron. And then finally, if you remember, I said that the siderophores that we find there typically have this fatty acid tail, and we thought that they were sort of either associated with bacterial membranes or anchored into the membranes, and that's how they gain their efficiency to make this a useful process. But if that were true, then what we'd expected was that most of those siderophores would be in the particulate phase, not the dissolved phase. And that turned out not to be true at all. So this is just one sample from the station 6. The bottom trace is the dissolved siderophores. There's 200 picomoles. It's mostly one of these marinobactins. We looked at the siderophores in the particulate sample that came from that water sample. Much lower concentrations of siderophores, and they all turn out to be these furoxamines, which we rarely see in seawater. So these ones with this amphiphilic tail, for some reason we find these dissolved in seawater. The ones that are actually quite polar, we thought would dissolve nicely in seawater, are all in the particulate matter. And we suspect that those are being used internally to move iron around the cell. So it appears that these things, most of the inventory, 96% in this case, is in the dissolved phase, and it's still these amphiphilic siderophores. So why are they at 200 to 400 meters? So what we did is we did a number of experiments at Station Aloha, which is a long-term ecological study site just north of Hawaii. It's near our Station 18 from the GP15 section. So the trace on the left here is the trace of nitrate plus nitrite, looking at the top 500 meters. So nanomolar concentrations in the euphotic zone. And then once I get below the euphotic zone, the concentrations go up very rapidly. So here's this zone from 200 to 400 meters. I've got tens of micromoles of nitrate. The plot on the right is from data from Jess Fitzsimmons. Here's now nanomole, 0 to 1 nanomole of iron, high concentrations in the surface because there's dust coming in the surface, hitting the surface, and iron dissolving. That iron is being rapidly consumed in the euphotic zone by microbes, which draw the concentration down. And then right below the euphotic zone, the concentrations start to rebound, but they rebound really slowly. We're going from a low of maybe 100 picomole to something like 200, 250 picomole at 400 meters, which is sort of this border between where iron becomes limiting or not. So our thought was, like I said, was that in this this twilight zone, this upper mesopelagic, you've got carbon substrate being supplied by sinking particles, nitrates there, phosphates there, but the iron hasn't recovered yet because the speed at which those particles release their nutrients as they sink is quite different between the different nitrogen, phosphorus, and iron. So we wanted to test that. So what we did is, in the lab, we grew some bacteria, made marinobactins, and we labeled them with iron 57, which is a form of iron that's very, very rare in the ocean. And so here's my trace of our spike that we're going to add to the samples. At the beginning of the experiment, you can see we have different marinobactins. This is now a trace of iron 57 in dark blue, and the light blue is there's a little bit of iron 56 in it. We just add those to seawater in bottles. We incubate them for a couple of days, and then we find out what happened to the iron 57. And so here's a death profile of what happens. So I should say that there are different siderophores in this, just A, B, and C. And so these plots all reproduce A, B, and C. And so what you're looking at is the amount of siderophore that was added, which is the dash gray line here. And then after 5 days of incubation, the amount of siderophore that's left in the sample. And you can see in the euphotic zone, and right to the bottom of the euphotic zone, most of the siderophore that we added is still there. It isn't taken up. But when we get to this 200 and 400 meter depth range where we saw a lot of siderophore production in the GP15 trace, all that siderophore is gone. It's been taken up. So this was all removed. And there's two ways that can happen. It could be just respiration. It could be that this is a labile carbon substrate, right, that we're adding. And bacteria down here are really interested in labile carbon substrates, whereas maybe not so much up here. So it could be that they're just using it to generate energy or biomass, or it could be that they're taking it up because they want the iron. And we reason that if they're after the iron, then that siderophore should be pumped back into the sample, but now it just doesn't have iron in it. So it's not going to show up in our analysis because we're just looking at the iron. And so just like I did in the equatorial Pacific samples, we took the extracts after we analyzed them and we added iron back into it. And that's what you can see here. So this is just one of those siderophore peaks. The blue hatch line, or the blue solid color, light blue, is the siderophore that was remaining at the end of the experiment. It's there at the surface and it's gone between 200 and 400 meters, then it comes back. The dark blue is the siderophore that reappears once we've added iron to the extract at the end of the experiment. And the gray is just the siderophore that's gone from the experiment. And we interpret that as this is just the siderophore that we added that was respired to CO2 and nitrogen. This is siderophore that was used to recover iron. And this is just the siderophore that was unused. So, you know, what's happening is we're adding iron 57, this little cartoon guy here, is being taken up by the cell. The iron is being removed, or it's being taken up by the cell. The iron is being removed. It's going back out into the environment. It can grab iron 56 when it's in the environment, or it could grab aluminum. And when we actually looked at the amount of aluminum 27 or iron 56 at the end of the incubations, they had increased slightly. So it showed that the iron 57 that we added was taken into the cell, it was removed, and the siderophore was put back into the environment. So this cycle was happening. It was a relatively short incubation. It's a couple of days, but low temperatures. But most of the siderophore that was lost, something like 60, 70% of it, was used for recovering iron. So it really demonstrates that iron's being, the siderophores are being produced and used down there to acquire iron. So as I said several times, what we think is happening is that the iron, you know, it's high in the surface, so they're not really iron stressed. When it gets to the bottom of the euphotic zone, all that iron's kind of gone. They become iron stressed. The green curve is the POC flux measured in sediment traps. So it's high in the euphotic zone, and this zone right below the euphotic zone, you have this really high POC flux, so high flux of substrate, lots of nitrogen available, but no iron. And so that leads to the production of these siderophores. And so we could use that iron to nitrogen ratio now to look at other ocean basins. So we haven't made any, well, that's not true. So we can predict where we should find siderophores based on nutrient data. So these are data from the, you know, South Pacific, Southern Ocean, the Eastern Tropical Pacific, that one that I showed you where we did our initial sampling, in the North Atlantic. And the hot colors are just very high iron to nitrogen ratio. The cool color is very low iron to nitrogen ratio. And the border here is 50. So in the Southern Ocean, where we know it's iron limited, in fact, we predict that we should find siderophores. In the South Pacific subtropical gyre, the iron to nitrogen ratio is high enough that we don't think that we should see any siderophores here. We should see them below the euphotic zone between this region of 200 400 meters. We think that's true for the tropical Pacific as well. This is the cruise track where I had the 4 or 5 samples, and right where it's blue here is right where the samples that we actually saw the high siderophore concentrations, very low inshore and offshore. But we also predict that the mesopelagic here should also have lots of siderophores. That's not true in the North Atlantic. The iron to nitrate ratios in the North Atlantic are high all the way through the mesopelagic. So we think that there should be no siderophore production down there. So what does that mean? We don't really know. There's only been a couple of studies, like I say, that have looked at iron limitation in heterotrophic bacteria. This one by Portel, and there's another one by Matt Church. But what they seem to show is that when bacteria are iron limited, it really slows the growth rate down. And there's some suggestion that it really impacts growth efficiency. So that's the amount of carbon that the bacteria takes up to build biomass, and it's the amount of carbon that's devoted to biomass synthesis versus respiration. And efficiencies go really down. So much more carbon is respired per unit of biomass than when you're growing under high iron. So we think both of these will really impact the way carbon is cycled in that upper mesopelagic zone. And that potentially, we don't know if it's true or not.
Sorry, Daniel, I'm really sorry to interrupt, but would it be possible to wrap up like
I have one more. Yeah, this is my last slide.
Okay, perfect. Thank you.
So it suggests that that biological carbon pump that people are depending on to get iron CO2 from the surface into the deep ocean might be much less efficient because of iron limitations. So anyway, these are the different thoughts we have on it. That the nutrient limitation, which we up till now had thought was only really in the euphotic zone, but it actually is much deeper than that. The region of the ocean that's impacted by iron is much greater than probably the 30% calculated by models. There's sort of unknown effects on this biological carbon pump. And the siderophore, this whole system is really one way, and it's quite common way for microbes in the ocean to get iron. So I'm wrapped up. Sorry. Ooh.
Thank you very much. And thank you very much for the great talk. We're just a little bit over time, but I think we have time for maybe 1 or 2 questions from the audience.
I will start. Daniel, thank you. It was a really cool talk, and thanks for making it so accessible. So I will have a non-expert question. I really like this idea of sampling water and then adding a little bit of iron and see how the amount of detectable siderophores, so those that bound to iron, increased.
Right.
And you mentioned something about the timescales at which this process happens. Can you elaborate a little bit on that? How do you know how much it takes for this additional binding?
Right. So we're still investigating that. So when we did the experiments, we just didn't know. We thought things would be very slow. So we did a 5-day incubation, and just for lots of sort of logistical reasons, we were limited because we had to get trace metal clean water and whatnot. So those incubations were done for 5 days, and they were done on deck incubation experiments. So the temperature was whatever, probably 20 degrees or something like that. And so it happened really, so they were gone. It could have been gone in a day. And we think that that might be true because we did do a time zero. And the time zero, so we add the spike and we immediately process the sample, but that processing takes about 2 to 3 hours. And in every case, the time zero was lower than the control by 20%. So we suspect that it happens at those temperatures within a day. And so I went back out again in August, and I should say we've done that experiment 3 times. and we always see the same results. And so then I went back out in August again, and this time I had in situ temperature incubators, and we did time points. And so now I'm incubating them at 13 degrees or whatever the temperature is down there. And I've got 1, 2, 3 day time points, and we haven't analyzed those samples yet, but we should know quickly. But our thinking is that happens probably within a day, or at most 2 days. So one last thing. So you can, if you know the rate, you can calculate the bacterial iron demand, right? Because we know how much iron is going into the bacteria, and we have a sense of bacterial productivity, and we have a sense of what the carbon to iron ratios should be. And at the moment, they're too high. So there's more iron being taken up by siderophores than the bacteria need. So either the temperature has, either the temperature effect's going to knock that down by a factor of 2 or 3, or there's a process called sort of luxury uptakes of nutrients. If you're nutrient starved and then your nutrient shows up, we know that bacteria, it will take up more than they need in the anticipation of running out in the future. And so we don't know which of those is going, but we should have a better handle on that in the coming months. Great question.
Thank you.
I see there is another question from the audience.
Hi.
Hi, Danielle. My name's Orkun. So we are watching you here as a group, actually. The several of us are here. And brilliant talk. Thanks a lot. So I wanted to ask 2 related questions. So if I understand correctly, iron 2 is readily soluble, whereas iron 3 readily precipitates.
Right.
So my questions are, in the early slides you had, the first slide you showed the sort of dissolved iron concentration in the oceans. So do they then distinguish between these 2 forms? And then the sort of related question is, I assume the siderophores, do they grab the iron 2 or do they sort of solubilize iron 3? And also in your measurements, how do you distinguish between siderophores and any solubilized iron?
Okay,
so maybe a bit too many questions, but
no, there's 3
they're
questions.
all related. Yeah,
I'll do the last one first. Because we can measure the mass of the siderophore iron complex, we can measure it to within 1 millidalton, which is the weight of an electron. So we can actually distinguish between iron 2 and iron 3 based on its mass. And it looks like it's iron 3. Also, they coordinate with the siderophores somewhat differently, and you would have to add hydrogen to do that. And so we're pretty confident that it's iron 3. People do measure iron 2 and iron 3 in seawater. Iron 2 concentrations are really low. It's not the stable form of iron. In surface waters, there is a photoreduction of iron 3 to iron 2. And there's been, I don't do it, and I'll be honest, I don't really follow the literature that much. It's just sort of what I get from talks and whatnot. But the lifetime of iron 2 in seawater, I think, is on the order of a few minutes. So the reduction occurs, and very quickly it's oxidized back to iron 3. And I think that nearly all of that occurs in the very surface layer of the ocean. There might be biological reduction of iron 3 to iron 2 that I'm not fully aware of in the subsurface, but the upshot is that the iron 2 concentrations are really low and the lifetime is really short. And then finally, yes, siderophores are built to complex iron 3. They're not built to complex iron 2. That's a different sort of geometry of complexation. And the stability constants, the sort of strength at which it grabs iron for iron 2 is much, much lower than iron 3. And so when that iron 3 is brought into the cell, there's a reductase there. In gram-negative bacteria, it's a reductase, not in gram-positive. There's a reductase that reduces that to iron 2, and then other ligands can pull that iron 2 out of the siderophore complex. And that's how in gram-negative bacteria it's thought that the iron is actually taken out of that complex, is through a reduction. So I got all 3.
Yeah, so the iron 3 is in a precipitate form, I guess, in some sort of granular form. And when you sort of look at these samples, do you then sort of pick this up as?
No, so in seawater, there's techniques they use electrochemistry, which are these anodes, and they reduce, they add chelators to seawater, and they look at the reduction potential, and they show that the iron 3 is actually stuck to the organic matter. Right. So siderophores are sort of one form of organic matter that microbes design and use to actually acquire iron. And the definition of a siderophore is it binds iron strongly, but it also, the key thing, lots of things bind iron strongly. It facilitates iron uptake. It makes the uptake of iron much faster than it would be otherwise. And so there's, that milligram of iron, the other hundreds of thousands of organic compounds that are in seawater, many of them bind iron. And you can see that when you look at our traces of our analyses. We have this sort of baseline in our chromatogram, where the peaks sit on top of. And when we get in, especially close to the surface ocean where there's the coastal ocean where there's lots of iron, that baseline there's a huge hump. And these are all these very complex humic substances and whatnot, which are binding iron. They bind it weakly, but they bind the iron. So there are mineral forms of iron, which are typically particulate. There's dust, right? And you can filter those out. You can look at that. And people have done experiments where they take dust and they add siderophores. And so, there are certain mineral forms of iron which are really, it's very crystalline and it's very hard to get the iron off of those. But siderophores can do that. They can actually dissolve crystalline iron oxides, and they know that happens. There's some nice video online where these trichodesmium, which are these colonies of marine algae or cyanobacteria, which have heterotrophs as with them, and they physically capture iron particles, and then the bacteria go to work on it, and they dissolve the particle down.
All right, thanks a lot.
Other questions? Sorry,
Yeah.
I went on so long.
Oh, no worries. Thank you very much, Daniel, for the great talk and for presenting at MOB. Thanks everyone for the very nice questions. And see you all next time.
Oh, I should just say that I had lots of collaborators, and I had really great support from the Simons Foundation and other people. This has been a long project, and it was many years of failure before we got it to work. But I want to certainly acknowledge all these collaborators and funding sources.
Thank you so much.
All right.
Thank you.
Thanks. Bye now.
Thank you, Daniel.